So the Jain Foundation has a very specific goal. That goal is to establish a first therapy for limb-girdle muscular dystrophy, 2B specifically. And ideally, we'd like that intervention to be something that can reverse the disease course, also known as a cure. I think all of you with the disease, that's what you want. You want to have this behind you and some return to normalcy. At a practical level, we're looking at anything that can improve the quality of life of patients that have this disease. And that means that in the drug development process, we may be able to repurpose some drugs that are already available that might not be a cure, but might be capable of slowing the disease down. And so the big differential there is time. And we prioritize things based on finding something soon. Obviously, there's a balance. If something's going to have very little effect and take a long time to develop, then maybe it's not so interesting. If something potentially has a large impact, though, then maybe it's worthwhile to pursue it, even though it may take a long time to develop. Alternatively, if there are things out there today that are FDA-approved drugs on the market that we can simply repurpose and apply, and that gives patients a benefit, well, even if it's a slight benefit, the fact that we can do it soon is a major advantage. So where we started. The foundation was established in 2005. At the time, there were a few very important discoveries. One is that the gene was known. And the other is that it seemed to have some sort of involvement in repairing muscle membranes. So when Plavi started, while those two discoveries were extremely important, what she found is that they were made by labs that were primarily studying other diseases. And this happens quite a bit in science. When you're doing science, you're exploring the unknown. And if you're going after a particular disease, you don't quite know what all the relationships are. So Plavi started working with the labs that made those discoveries and rapidly found that there was a vacuum in terms of leadership in the field. And what I mean by that is that because their primary focus was on something else, it was not on dysferlinopathy. So that left the foundation as the central resource to take leadership in the field. So having made that observation in 2007, Plavi decided she needed additional help at the foundation, expanded its personnel by hiring Laura and I. And we took off with the express purpose of being leaders in this field and trying to find a true therapy for our patients. So having recently entered the foundation, the first thing that Laura and I did was assemble our desks, which we recently bought from IKEA. The second thing we did was make an assessment of the field and try to figure out, okay, what does it take to actually get a drug to market? And our first source of reference would be drug companies. That's what they do. They produce drugs and bring them to market. And when you talk to drug companies or look at their websites, you rapidly find these sorts of models that demonstrate what they feel is the drug development process. There's usually quite a bit of propaganda that's associated with these websites, and they want to impress upon you that the price they charge you for the drug is really worthwhile because it takes so many years and X million dollars to actually get the drug to market. If you do look at some of these websites, don't be too discouraged by their numbers. It is a little bit of propaganda there. It's okay to say that it's difficult. It does take quite a bit of effort and typically many years of research. So in looking at these paradigms, what we found is that even beginning to approach this process, we had some deficiencies. So the first step is basic research. And there's a scientific term there that Sarah always harps on me. What is basic research? I mean, is that different than complicated research? I mean, what are we talking about here? So basic research is a term that's used to describe research that's typically done at academic universities where you're just trying to understand the disease or look at some fundamental part of the process to establish a knowledge base. When you talk to drug companies, what you find is that their idea of basic research assumes that you already have a fairly extensive knowledge of the disease process and that it's sufficient to ID drug candidates. As we started out doing research on LGMD2B, we realized that that was an area that we sorely needed to improve. The next step is preclinical studies. So there's another kind of scientific term. What does a preclinical study mean? Typically in drug development, that means testing your therapy in an animal model. So that's a prerequisite to testing the therapy in a person, because obviously the FDA wants you to make sure that your drug actually does what it's supposed to do, and B, that it's not going to hurt anybody. But to do a preclinical study, that means that you have to have an animal model, and it also means that you have to be able to track the disease in that animal model. As patients who have the disease, that might seem somewhat simple. I mean, you understand what's happening to you, and you experience it every day. But when you're a scientist and you're looking at a mouse in a cage and you're trying to figure out whether the disease has progressed or not, it can be quite a bit more difficult. So that was another point that we had to establish, is how to look at this disease in animal models. Likewise, the next step is doing clinical studies on patients. When you look at this in detail, this also assumes that patients are identified and ready to start in a clinical trial, and that methods to study them have been established and are capable of picking up a difference if your therapy works. So having looked at this and talking to drug companies, we realized that there were a number of deficiencies in the field that we had to fill. In addition, we found that pharma, while they have essential infrastructure for developing drugs, including the manufacturing capabilities and marketing and dealing with regulatory agencies, they're pretty much driven by one thing, which is profit. And to them, it comes down to a very simple formula. The drug price times the number of patients they can sell it to has to far outweigh the possible developmental costs. So, well, that throws another wrinkle in the mix, in that we realized that drug companies would not be interested in our disease until we got sufficiently through the process so that the costs that they would have to put in to reach the end were reduced to the point where the financial equation made sense to them. So having figured all this out, it basically gave us a roadmap of what we needed to do next. We needed to build a knowledge base. Understanding a disease is essential for building a therapy, and you see that in many aspects of drug development. So some of the fundamentals we had to establish. In the early days, we found that the scientific field itself needed tools to simply study the disease, whether they're mouse models, cell lines, DNA clones, antibodies. We need to determine what goes wrong in a muscle when dysferlin is missing and the possible intervention points. And we needed to find symptoms related to the disease so that we could track its progression, either in cell lines, animal models, and even in people. So our philosophy moving forward was to pursue all options. We had a conference where our theme was leave no stone unturned. Learn from other diseases when possible, and focus on funding dysferlin-specific issues that are related to finding and testing therapies. So I'll save you all the details, but after several years and quite a bit of money spent, we have a much better situation today. So our progress toward building a therapy, the Jain Foundation now acts as a central distributor of research tools for academic labs as well as well, anybody who needs them. Our research program has identified multiple intervention points that are being evaluated for their therapeutic potential. Symptoms related to Dysferlin's loss have been identified in cell lines. I know this sounds a little, why do you care about a cell? But being able to study cells allows you access to high throughput screening platforms where you can literally look at thousands of drugs to see if they have an impact on the disease. So it greatly expands our ability to look at different therapies. Symptoms related to Dysferlin's loss have been identified in mice, and we now have a platform where we can study disease progress in mice and have a shot at reversing them. And critically, we have a growing number of patients that are genetically diagnosed and are clinical trial ready. That has taken a long time to develop, and I can't emphasize how much work it has been to essentially buck the system and make sure that patients are not just diagnosed as having LGMD, but go all the way through to genetic diagnosis and have their mutations known. With those patients in hand, we were able to start our clinical outcome study, which is underway, and that is designed to give us the tools we need to run a clinical trial so that we can track disease within the patients and have a chance of seeing whether some of these therapies are actually giving a benefit. So with progress in these key areas, we now have partnerships with a few different pharma companies. I'll say the interest is not great at this point, but the fact that they're paying a little bit more attention to us and listening and actually providing us with reagents and sometimes drug is very encouraging. So to go back to our timeline, in 2007, we were assuming leadership of the field. In 2010, we had established enough information that we could accurately study animals, and through Jain Foundation funded research, we're able to show that Dysferlin gene therapy in mice could be effective. In 2012, we started a program where we are translating some of the discoveries that we make in academic labs to what are called CROs, or contract research organizations. And this is a process that's similar to what kind of mid-level pharmaceutical companies use. It's rather than develop and incorporate all the infrastructure they need to do preclinical trials in-house, many pharma companies outsource it to companies that specialize in those techniques. So we've done the same as a step towards moving things closer to what pharma would be interested in. And we've now had projects with, I believe, 5 different CROs, some of which are still ongoing. In 2012, we started the cost study, which I hope many of you are familiar with. And that's going to be critical towards our future steps in having clinical trials. In 2013, we initiated a biomarker study. A biomarker is another kind of technical term. Basically, biomarkers are any sort of biological way of measuring a person that you can use to track the disease. In this case, we're looking at patient blood. So what we're trying to do is identify things that are in the circulation of our patients that might track with their disease progression. That study is to be completed in 2015. In 2014, we started our first preclinical drug trial at a CRO. I think of that as a very important step. Not all the drugs that we're going to test are going to work, but at least we have a way to test them now. And in 2018, we plan to have costs completed, and then we should be in a better place to start running large-scale clinical trials. In large scale, everything's relative. We have a small patient population, so larger than some of the clinical trials that have occurred to date. So with that, I want to get into the different emerging therapeutic options. Like I mentioned in the beginning, this is kind of broken down into two parts. Part one is looking at therapies that are able to slow down disease progression. And like I mentioned, there's pros and cons here. The pro is that repurposing approved drugs may be very rapid. The con is that many of them are targeting some part of a disease process, and they may only provide a mild benefit rather than a true cure. Part two will focus on more recent technologies that allow us to put dysferlin back. The pro is that this is conceptually simple. LGMD2B is a disease that's caused by the loss of a particular protein, dysferlin, and so conceptually, why not just put it back? In terms of the biology, it makes sense. If you return it, it should do what it was supposed to be doing in the first place, and all the processes should return back to normal. In practice, it's a bit more difficult. Many of the issues revolve around delivering dysferlin and doing it in a safe and effective manner. The interesting part about these technologies is that they rely on our relatively recent understanding of the genome and how it works, and how a mutation in a gene gets translated into making the protein, and where in that pathway you can intervene. Because of this, regulatory agencies aren't as adept at dealing with this kind of strategy, and it's been a little bit of a struggle to get them to realize what the issues are and how this is going to move forward. So they're adapting to the science in this case. So to get into part one, this is going to focus on slowing down disease progression. So like I mentioned in the first part of the talk, one of our research focuses was trying to understand why the absence of dysferlin leads to muscle loss. And the key thing in understanding that process is to identify points where we can intervene in it. I'm not going to go into the process in much detail. But as we go through these, you'll see that at each step, we've identified areas where we think we might have a chance to develop a therapy that could have some benefit. For example, dysferlin is thought to be involved in membrane repair. So if you can do something that stabilizes the membrane so it's less susceptible to damage, then possibly that could have some benefit for the patients, because they wouldn't need dysferlin to be there as often. Likewise, proteins that can replace dysferlin and its function in membrane repair could be a viable option. Once a muscle fiber is damaged, there's a whole process by which the muscle fiber actually dies. There's research that indicates that calcium levels within the muscle are critical for that process. This gets a bit technical, and there is a whole area of science that revolves around this. It's the calcium that's used in this process isn't something that you can change by simply drinking less milk or doing something dietary. It's part of how the cell processes calcium. But within that, there are some opportunities to maybe change the regulation and alter that pathway. Likewise, once a muscle fiber starts dying, there tends to be an immune response that happens that normally should happen, that's involved in clearing out the damaged tissue and facilitating recovery. There are aspects of that process that can induce more damage in the muscle. We are exploring that field actively to see what sort of opportunities are available. And finally, you have the ultimate situation where you have a loss of strength. And there is significant interest by pharma and chemical entities or drugs that will simply increase strength for a lot of different reasons. So that brings up the question, can you simply kind of outgrow the problem? Give the patient some entity that will allow their muscles to grow faster than they're breaking down, and essentially tip the balance in the other direction. So I'm going to go through these one by one and tell you kind of the state of where things are. So with membrane stabilization, we've had some success with that in artificial systems. We've never found anything that had any benefit in animals at this point. It's an area that we continue to pursue at a basic science level because it seems like it's fundamental to the disease process, but it's not maybe one of our major focuses. We do have a very keen interest in immune modulation, and part of this is because there are a lot of approved drugs in the immune modulation field. Some examples are rheumatoid arthritis drugs that target autoimmune disease. And that field is rapidly expanding. So the number of targets that are available is increasing. Our focus is on the need to selectively prevent the damaging effects of the immune system without interfering with the beneficial effects. So there are some issues to tease out in that regard. At this point, we've tested some of the best options in preclinical trials and in collaboration with a couple of pharma companies. To not reveal too much, we have yet to see a home run in this department. In exploring these options, we have had some issues where access has been blocked by the companies that own them. This is a problem that we're starting to get into, in that a lot of the immune system modulators are what are called biological drugs, which means that they aren't just chemicals, but they're proteins that are made in cells and manufactured and then given to patients. So it's a much more complicated manufacturing process, and they tend to be quite expensive. And just the expense of them limits our access. And on top of that, that means that we need to approach the drug companies and get them to provide the drug. But sometimes they don't want to do that because they're already selling it for a different indication. And if we start testing it and find something that's unfavorable, then that might ruin their profit stream. That situation can be a little bit frustrating, but it's part of working in the field. And last point, academic studies are attempting to refine what aspects of the immune system are critical for, or that we do want to target for benefit. Let's see, then we have repair-mediating proteins and drugs. There was a time when we had a main therapeutic candidate. We were able to examine it thoroughly enough to demonstrate that it no longer looks very promising. Additional candidates in this area aren't obvious, but in our basic science studies where we're trying to understand membrane repair, we're keeping an eye out for anything that might emerge as another potential candidate. Calcium regulation. So this is kind of an interesting field in that there are quite a few drugs in the cardiovascular field that have the potential to be repurposed. Again, we've been able to put a couple of the best options into our preclinical studies in animals. Those are ongoing, and we are continuing with the academic work to refine the contribution of calcium in this process and establish whether manipulating it is going to give us some kind of benefit. The final point in part one is looking at increased regeneration. This is an area of science where pharma has a big interest in something called sarcopenia, which is age-related muscle loss. And there are a couple of drugs, they're based on myostatin inhibition. A few of you may have heard of that, which have really dramatic effects on the ability to build muscle. The question that we have is whether those sorts of therapies would actually be applicable to something like muscular dystrophy. And we have some basic science studies in animals ongoing that we hope will establish whether, if pharma gets those drugs approved and on the market for sarcopenia, if they might be useful to repurpose for LGMD2B. All right, so I'll move on to part two, which is about putting dysferlin back. So a bit like the disease process where we wanted to intervene in the pathway between the absence of dysferlin and the loss of muscle. In this section, we're going to look at ways that we can intervene between the presence of specific DNA mutations and the production of the dysferlin protein. So the first step, and one of the ones that I think gets talked about quite a bit, is Stopcodon Readthrough. So how many of you are familiar with what your mutations are in dysferlin? And I realize that this is going to dive a little bit more into the whole science side of things and how a gene ends up making a protein. I think the bottom line here is that there's no way around kind of describing what these technologies do without discussing some of this stuff. And there is a level of importance that I want to impress upon you all that, a lot of these emerging therapies, you really do need to know what your mutations are, because some of these therapies are only applicable to certain kinds of mutations. Stopcodon Readthrough is definitely one of those. In this case, you'd have to have what's essentially a point mutation in the genetic code that leads to a signal that causes the protein production to stop. If you can read through that and make the protein again, then effectively you could restore dysferlin protein production in a patient that has this kind of mutation. Another technical term and possible therapy is called exon skipping. So if you talk about a gene in your DNA, the gene tends to be fragmented into pieces that are all reassembled and make the code. Those pieces are known as exons, and there is some ability to alter which exons are present in a given message to make a protein. There's interest in the field in some things called chaperones. So chaperones are either drugs or small proteins that help facilitate the folding of other proteins. And one way they can help is that certain mutations in dysferlin may lead to its misfolding. So it's basically an amino acid change, and it has to fold together in a certain way to make a functional protein. Chaperones can help facilitate that process, and there's some interest in therapy development there. There's a lot of talk, and it's kind of a hot field right now, looking at gene editing. It's an interesting emerging technology. It's probably not something ready for therapeutic development And then there are a few other strategies where you might just be able to put dysferlin back. One is protein therapy. Why not just make a bunch of dysferlin in a vat and inject it into people and see what happens? Gene therapy, which is generally delivering a good copy of the dysferlin gene, typically through a virus, into the cells so that they have a copy that they can make the protein from. And another approach is to use stem cell therapy, where you would take cells that have a good copy of dysferlin in them, put them into the body, and they should integrate with the muscle, or hopefully will integrate with the muscle, and then supply dysferlin back to it. So to discuss these individually, I kind of broke them down into two different sections. The first is looking at things that in their ideal state would be permanent and irreversible. This is a little bit of a double-edged sword. It's great that some of these therapies could be permanent and that you would only have to go in for essentially be treated once and not return. The issue there is getting them through the regulatory agencies is a little bit more strict because they are irreversible. So while this is in development and we're working it out, the FDA, for good reason, is taking a conservative approach, because once you give these kinds of therapies to somebody, you can't really take it back. So if you didn't get it quite right, there's not much you can do about it. So for gene therapy, AAV gene therapy is pretty much the state of the art. We're very happy to be working closely with Dr. Mendel on looking at AAV dysferlin gene therapy. I'm not going to go into this anymore because he's going to be talking after me. And we'll give you much more detail on that process and where it stands. So gene editing is kind of a theoretical field. It's been garnering a lot of attention. You may have heard of like CRISPRs or Cas9. There was an incident that made it onto CNN where Francis Collins mentioned that the NIH would not fund any genetic editing of embryos. And that was in response to a study that was released from China. It's a very interesting field because the technology is emerging where we might be able to literally go in and change the DNA error that you have in a gene. At this point, it's not really a therapy. It's being used to develop tools for research primarily, whether they're cell lines or animal models. The efficiency is rather low, and that's one of the major reasons why it's not being developed into a therapy. It brings up major ethical considerations. There's a lot of concern about what it means for mankind to be able to start altering our own genetic code. That debate is mostly, not entirely, but mostly limited to the gene editing field, because unlike some of the other technologies, you do have the ability to potentially also edit germ cells, which would mean in a case like this, you could prevent the chance of passing on the gene to your progeny, which is a good thing. But it also then becomes a very slippery slope, and that, where do you stop? If you can do this for this gene, can you do it to make, your kid's eyes blue? Can you, so that's going to take considerable ethical debate. We aren't actively funding anything in this field, but it's a hot area of science right now. It has plenty of funding behind it on its own. So we're just watching the technology to see how it develops. Move on to stem cell therapy. So stem cell therapy has been successful in proof of concept studies. So generally these are studies in mice where people have shown that we can do this. The problem with stem cell therapy right now is that the efficiency remains too low to really be an effective therapy for people. And the major hurdle is in actually getting these stem cells to the muscle and engraft so that they do something useful. In terms of our view of the stem cell field, we had been active in it previously for a number of years in some projects where we were addressing dysferlin-specific issues. We'd gotten to a point where we realized that the major issue that needs to be solved is not dysferlin-specific. It's really is central to kind of this, what I think of as cellular engineering, where we're just beginning to understand what makes a stem cell and how we can manipulate their properties. And there is quite a bit of funding in that area as well, and it's not really an area of science that we have the capacity to move. So at this point, we don't have any active projects, but we are keeping a close eye on this field because at some point there could be a breakthrough that we'll want to take advantage of. So the second part of putting dysferlin back addresses potential therapies that are transient and reversible. So this is the flip side of the other coin, and that being transient therapies, they may not be initially as attractive because they're things that you would have to take for life. And it could be inconvenient to take a pill every day, but you know, that is what it is. But they're also reversible, and this gives them a little bit of advantage, apparently, with the regulatory agencies. So some of these technologies have been moving a little bit faster through the FDA and the EMEA. The first is Stopcodon Readthrough. So there is a drug, Adalurin, that is conditionally approved for use in Europe and is on the market in Germany for Duchenne muscular dystrophy. This is, I mean, it's really interesting in terms of, well, in a lot of ways. I guess some of the issues that we're looking at is, you know, while it's approved for Duchenne muscular dystrophy, we're kind of treading new ground in terms of the regulatory agencies and how to apply this, because this particular drug could be used for just about any genetic disease, provided the patient has a stopcodon mutation. But the regulatory agencies are set up to study particular diseases, not whole suites of diseases. So it'll be interesting to see how this ends up being applied across multiple diseases. So far, the benefit to Duchenne patients has been relatively small. This is mainly thought to be because the drug itself has a limited efficiency. So it's only reading or only allowing read-through of the stop codon, I mean, at a maximum about 10% of the time, which means that the patients are making between 1 or single-digit percentages of the normal amount of dystrophin they should have been making. We don't know if that level of dysferlin is going to provide a benefit to patients who don't have any, but we won't know until we try. There's also an aspect here where this drug is only going to be applicable to patients with nonsense or stop mutations. If you don't have one of those, it simply isn't relevant. Exon skipping is also moving forward and showing quite a bit of promise in Duchenne muscular dystrophy. We have a research program where we're assessing its applicability to dysferlin. Here again, in this case, you're not looking at a specific drug per se, but more of a technique. The technique's fairly robust, and it seems like it could be applied to dysferlin. The problem is we don't know whether we can skip exons in dysferlin. In dystrophin, they know that the main important parts of the protein are on either end. And so if you have mutations in the middle, you can skip chunks and the whole thing skips back, or the way it's made, it doesn't really matter that they're missing because you still have the two important functional ends. From what we know about dysferlin, we don't think it operates like that. Nevertheless, this technology is moving forward rapidly enough to the clinic for Duchenne that we feel like it's important to address and determine whether exon skipping is going to be relevant. If it is, it will only be applicable to patients that have mutations at specific points in their gene, because those are the exons that will be able to be skipped. At this point, we don't know which can, so we don't know where those mutations would need to be, but the point is that it again will have a limited patient population for which it will be relevant. So that brings us to chaperones. Their proof of concept studies with chaperones have been successful, but it's been limited to a single specific point mutation. There is one drug that's available, Velcade, and there was a study in Switzerland that demonstrated that it had some potential to treat, I believe it was 3 patients with one very specific point mutation. There haven't been any studies to show actual efficacy, so we don't know whether it's allowing enough dysferlin to be made to really have a benefit for the patients. But that's in limited clinical trials in Europe. And one of the really limiting things here is that we've been collaborating with the researcher who's doing those studies, and finding patients that have this specific point mutation is very difficult. It's a very rare mutation that they've been able to identify as applicable. All right. So the final one is protein therapy. There are a number of technical reasons why we thought this might not work. We did fund a study to look at or establish proof of concept here. It failed in animals. Turns out you can make a bunch of dysferlin and put it in a dish, and it does improve membrane repair. But when you put it into an animal, getting it to the right spot so it's where it needs to be is a real issue, and the protein gets degraded very rapidly. So we've abandoned that approach for now, but we'd certainly look at it again should we find some other way to deliver the protein effectively. So the road ahead. I've given you a lot of different options and talked about them briefly. None of them are readily available to you right now. And in fact, no single emerging therapy is going to be a sure thing. There's a lot of different issues that are related to each of these, and it's tough to make a bet on which one might arrive to the clinic first. Because of that, we will continue to pursue them all, because we feel like that's the surest route forward. There are several therapies that are reaching the clinical trial stage of development, and as a patient who wants to help in this process, enthusiastic and committed participation is necessary to move them forward. So if there's something you can do, it's keep your eye out for clinical trials or respond to requests or suggestions that the Jain Foundation sends to you. And then, new therapeutic opportunities will continue to surface as we continue our basic science program and learn more about this disease. And really the great thing about where we are is from the beginning of the talk, we now have so much infrastructure and things in place that we feel like we're in a position where we can really start to address whether these therapies are going to be relevant. So I think we're in a great place going forward.